We continue the discussion on steps in gene cloning. The fourth step is transformation of recombinant DNA into the host cell, and the last step, screening to select the host cell that carries target gene.
Now we move on to step 4, which is transformation. Transformation is the process to transfer the recombinant DNA into the host cell. So you note that the host cell, here we are using the bacterial cells, so this cell must not contain any plasmid inside them. So all the host cell has no plasmid inside them. During transformation, in this condition we are going to use electric shock, so the host cell will be shocked and then it will take the plasmid. But also, in this process, you cannot control the process. Some of the host cell will not receive any of the plasmid. Some cells will receive the self-ligated plasmid. Some cell will receive the wrong recombinant DNA molecules. And some cell will receive your target recombinant molecule. So during transformation process, there are 4 possibilities. Number 1, untransformed bacterial cells, which does not receive any plasmid. Number 2, the host cell receive self-ligated plasmid. The third possibility is that the bacterial cell receive the wrong recombinant DNA molecule. And the fourth possibility is that the host cell receive the target gene that you want. So the 3 types of host cell here, we are not going to need them, but we just only need this cell. But we cannot see it with our naked eyes. That's why we need to move on to the last step, which is screening. The simplest screening process is to culture them in a medium containing antibiotic, for example, ampicillin. So we spread the bacterial after the transformation process evenly onto the medium. And then we incubate for a period of time, and after that you get a colonies of bacteria. So this is the technique to spread the bacterial onto the medium containing antibiotics. It is conducted under sterile condition. So the bacteria that is being spread on the medium, okay, some of that does not contain any plasmid. So this host cell cannot live in a medium containing antibiotic since it doesn't receive any plasmid. Because we know that the plasmid characteristic, it must have selectable marker genes, for example, ampicillin resistance gene. So when the host cell doesn't receive any plasmid, it doesn't have the ability to live in a medium containing ampicillin. So this host cell will die. This is the simplest way to eliminate the untransformed cells. The other host cell, whether it received the self-ligated plasmid, the wrong recombinant DNA, or the correct recombinant DNA, can live inside this medium and it will divide to produce a colony. So on the medium, you can only see the white colonies. It cannot be differentiated whether it contains the self-ligated plasmid or the right recombinant DNA or the wrong recombinant DNA. The next screening is to conduct the blue-white screening. The blue-white screening is conducted to differentiate the host cell that contains the plasmid that is self-ligated or the host cell that received the recombinant plasmid. The self-ligated plasmid containing cells, it will be blue in color, whereas the host cell that received the recombinant plasmid will be white in color. So the one that you want is the white colony, because the blue colony is the host cell that received the self-ligated plasmid, no recombinant DNA molecule. That means you eliminate by determining the color of the colony. The multiple cloning site of the plasmid is actually located in the LEGZ gene. So when you cut the restriction site in the multiple cloning site, the plasmid become open, and then when it combined back without any insertion, the LEGZ gene is not
disrupted.
So it is still functional. Whereas in this case, when you cut the restriction site in here, so when it receives the gene of interest, it will cause the additional base sequence at this region, which causes the LEGZ gene to be
disrupted.
That means this LEGZ gene has undergone mutation. So this LEGZ gene is non-functional. So when the self-ligated plasmid contains the functional LEGZ gene, it can produce certain enzyme that can digest molecule, namely X-gal. So the X-gal can be hydrolyzed to form a product. It will causes the colony to be blue, whereas this one is the one that we want. The color will be white. So you can see here, the selection is based on the color. The white colony containing the recombinant DNA molecule, whereas the blue colony containing the self-ligated plasmid. The last screening is to conduct the nucleic acid hybridization, which will not be discussed here, but only the brief explanation of it is that it will develop a film. So once you develop the film, you go back to the original colonies and trace it back. So we know that this colony actually contains the correct recombinant DNA. So there goes the 5 steps of the gene cloning.